Hello everyone, this is Dr. Pooja, your original guru of biochemistry. In today's video, we are going to study the alternative pathways of glucose oxidation. So far in the videos on carbs metabolism, you have seen that we have discussed the major pathways. Okay, and we all know that carbohydrates, being the universal sources of energy, they play the most important role in our body, isn't it? Because excess of carbohydrates can be converted into fats and they are stored, right? So now in today's video, we are going to study the alternative pathways of glucose oxidation. First of all, we need to know the difference between the major pathways and alternative pathways. As it is clear from the word itself, major means most important. I am not saying alternative pathways are not important. But yes, the basic difference is that major pathways are occurring all the time, right? And they are restricted almost to every tissue and organ of the body. But whenever we talk about alternative pathways, that means they have a significant role in most of the important organs, or you can say most of the organs of the body. So if we talk about alternative pathways of glucose oxidation, right, there are two pathways. One is HMP Shunt, right, and second is uronic acid pathway. Uronic acid pathway, right. Now first of all beta, you people should know what is the difference between a pathway, a cycle and a shunt, because these are very basic things. They appear to be very simple ones, but you know they play a very important role in our body itself. So if I talk about a shunt, what is a shunt? It does not have an end product. Okay, so shunt does not have an end product. Ab that doesn't mean that it will not finish. Okay, you must be thinking if anything does not have an end product, it will keep on going. No, it is going to form a product which will merge itself to the major pathways. So ultimately it is happening in the body for some relevant purpose. And once the purpose is over, it is going to merge itself to the major pathways. Okay, so always remember the shunt does not have an end product and it merges itself into the major pathways. Very important. Right. What are pathways? Pathways, we know, like this they are going. What is a cycle? Cycle, you know, just like the wheel. Like this, the cycle keeps on going. So, abhi tak so far in the metabolism of carbohydrates specifically, we have studied one shunt which is RLS, right? Rapoport-Lubring shunt, which is operative in the RBCs and which plays a very important role in our body to survive the anoxic episodes, which we have already discussed in detail. And we know that even though the net gain of energy is zero, then also RLS plays a very important role. Just suppose if we go for any trip to high altitude, just summer season is there, most of the people are heading towards Leh Ladakh. So what is the mechanism present in the body which keeps us, because normally you must have heard that say we have to take the Dimox, acetazolamide when we are going on high altitude. So there is some mechanism which is going in our body. And that is what is that Rapoport-Lubring shunt, which becomes more when we go over in a high altitude. A second shunt which we are going to study is HMP shunt, which plays again a very important role, right? And these are always, remember, the alternative pathways of glucose oxidation. Now, first of all, we are going to take this HMP shunt in detail. After that, we will go over to uronic acid pathway, right? So, abhi just now I have told you what is a shunt which does not have an end product. Now, the question is, this HMP shunt is actually known as hexose, monophosphate pathway, but the correct way is to name it as a shunt. Then it is also known as PPP. PPP, pentose phosphate pathway. And it is also known as Dickens-Horaker pathway also, as it was discovered by him. Dickens-Horaker pathway. Right? Now, first of all, since it is an alternative method of oxidation of glucose, what is the site where it occurs? What role it has to play in the organs? So, if we talk about the site of HMP shunt, the most important one are obviously the liver, because we know liver is the, again, kitchen of the body. Then it plays a very, very important role in the RBCs. It plays an important role in the adipose tissue. It plays a very important role in the lactating mammary glands. And it plays an important role in the skin. But the most important role is played in the liver, RBCs, adipose tissue. Very, very important. Okay. Then another important point is that it consists of two phases. And what are those phases? The phases are, one is a oxidative phase. Right? One is a oxidative, mane oxygen will be required in this, because whenever we talk about oxidation, oxygen, so oxidative phase. So oxidation or oxidative phase. And second is non-oxidative phase. Right? Then the question is again, site ho gaya, phases ho gaya, then basically what is the site and what type of process this is. Okay. So always remember, if we talk about the site, it occurs in the cytosol and it is an anabolic process. Right. So I can just write site means organelle is the cytoplasm. Very, very important again, and it's an anabolic process. Now, so far you must have seen the different videos on carbs. You know, whenever a process is anabolic, that means insulin is always going to promote that process. That means if I talk about HMP shunt, what is the role of insulin in this? Insulin is always going to promote this process. Okay. Now, coming over to this, these are the important points. Then coming over, what happens in oxidative phase and what happens in non-oxidative phase. And then we are going to discuss what is the significance, because if so many things are there, you know, so there has to be some significance attached to it. Now, before starting up with this, most of the students get confused between again the spellings, because there are so many, you know, different terminologies in biochem that it is but obvious. There is a confusion, but again, if you are confident about your things, if you know what you are reading, what you are thinking, then you can easily, you know, solve your things. Here, the students get confused between HMP and EMP. There is only a difference of H and E. But if we talk about the differences, if we talk about the functions, there is a world of difference between HMP and EMP. So, first of all, I'll just tell you HMP and EMP mein what are the basic differences. So, difference between, because it sounds familiar, no? HMP and EMP. Now see, HMP, abhi just now we are discussing, EMP is nothing but your glycolysis. So simple. Now why it is known as EMP? Because it was discovered by Ambedon, Meyerhoff and Pranas. So it is also known as Ambedon-Meyerhoff-Pranas pathway. But if we talk about the differences, you can yourself see this is HMP and say this is EMP. No doubt, both are occurring in the cytoplasm. Okay, so it is not a difference, but yes, if we talk about this is a anabolic process and this is a catabolic process. We all know this, right? And if we talk about the energetics, because anabolic hai, since there is no production of ATP, so no ATP is produced in HMP shunt. Whereas we know that if we talk about aerobic medium, so ATP is produced. We know that in aerobic medium 7 ATP or in anaerobic 2 ATP are produced. Okay. Then third important feature is that here carbon dioxide is always produced. But in EMP no carbon dioxide is produced because there is no use of carbon dioxide there. So these are the three main differences between HMP and EMP. Okay, because sometimes if the question is asked like this, write a short note on EMP. So, out of excitement, one knows that MP is there, then you'll start performing. No, there is a difference. EMP is glycolysis, which is altogether a different process. And HMP is a shunt, which is altogether a different process, which is strictly anabolic. And now the question is, if there is no ATP produced, what is the significance of this? That is what we are going to study now. Okay. So, and it is a multi-cyclic process. This can be also one of the differences. We can say multi-cyclic process. What is multi-cyclic? That it always starts with 3 molecules of glucose. Whereas if we talk about this, yahan pe to we always start with 1 molecule of glucose. So this is the main difference. 1 molecule of glucose, which undergoes oxidation anaerobically, aerobically, like that. So first of all, let's go through the oxidative phase of this. So I will just explain the oxidative phase of HMP shunt. Now see, this is the oxidative phase of HMP shunt. Okay, you can simply say that this is glucose 6-phosphate. Right, I'll just write 3 molecules of glucose 6-phosphate. It starts because it's a multi-cyclic process. And if you see what it is forming ribose 5-phosphate. So if I say 3 molecules of glucose 6-phosphate are there, so I can say 3 molecules of ribose 5-phosphate will also be produced. Now if we clearly look at this oxidative phase, we can simply say that what is happening? You can see carbon dioxide is being liberated. That is what I told you, that CO2 will always be produced in HMP shunt, right? And the second important feature which you people can see here is NADPH. It is not NADH, it is NADPH. Okay, most of the students keep asking, what is the difference between NADH and NADPH? Dekho, if we just look at the words, P is there and this P makes the most important difference. P is nothing but phosphate and we know that phosphate is a high energy compound. Okay, because again biochemistry is simply chemistry. What all you have studied in chemistry, the things which are occurring in a body comes under biochemistry. So phosphate beta kya hai? Wherever phosphate will be there, it will always give energy and that is what a body requires. Whether the body has to synthesize something or whether the body has to degrade something. So this is important. Okay. So you can see, you can notice 2 things here. One is that NADPH is being produced, right? And CO2 is being liberated. Okay. So I can say that NADPH will be produced and CO2 will be liberated. And we can also see ribose is also being produced. Ribose, which has how many carbon atoms? 5 carbon atoms. Right? So, in the oxidative phase, वैसे भी we know oxygen is there. In the presence of oxygen, reduction will be there. So, NADPH will be produced, and our main focus is on this particular enzyme, which we will be discussing in detail also. The name of this enzyme is glucose 6-phosphate dehydrogenase. Okay. So, I can simply summarize oxidative phase as, since it is a multicyclic process, I can say that 3 molecules of glucose 6-phosphate via oxidative phase will give 3 molecules of ribose 5-phosphate, liberating CO2 and generating NADPH. Okay, so I can write it also that oxidative phase liberates CO2, right? Synthesizes NADPH and synthesizes ribose sugar. And if you clearly see that there are how many NADPH here also and here also. Okay. So, here also NADPH is there. So, if I am saying 3 molecules of glucose 6-phosphate are there, so you can simply say 3 and 3. So, it will double up. So, it would be like 2, 2, 2. So, it would be 6 and 3 carbon dioxide will be liberated, right? And 3 molecules of ribose 5-phosphate will be produced. That is what happens in oxidative phase. Now, the question is, I told you it is a shunt. Shunt का मतलब क्या है कि it is going to form something which will merge itself. So, what will happen? 3 molecules of ribose 5-phosphate will intermingle with each other and they will merge themselves into glycolysis. So, second phase becomes our non-oxidative phase. Now, if you look at non-oxidative phase, I'll just show what happens in a non-oxidative phase. See, this is non-oxidative phase. See, this is ribulose 5-phosphate. Okay, it is forming what? Simply, it is combining with other things. It is forming xylulose 5-phosphate, ribose 5-phosphate. Or if we talk about the net result is this: fructose 6-phosphate, and you can say glyceraldehyde 3-phosphate. Or it will merge itself to form this. Okay. So, if I have to summarize this, let me just write. So, or we can simply say that if I have to elaborate it further, I can simply say that this ribose 5-phosphate, there are 3 molecules. Okay. So, I can say that 3 molecules of ribose 5-phosphate, 5 carbon atom and 5 carbon atom will be there. Okay. It will be xylulose also, I can say, because xylulose is also having 5 carbon atoms. So, what happens is this 5 carbon atom will combine with each other. So, 5 plus 5 is what? 10 carbon atoms. It will split up into 7 carbon atom and 3 carbon atom. Right? 7 carbon atom is pseudoheptulose and 3 carbon atom is glyceraldehyde. Okay. And this 7 carbon atom, again it will combine with 3 carbon atom to again split up into 6 and 4. And this 5 and 4 will then condense with each other to form, again 5 plus 4 is 9. So, it will split up to form 6 carbon atom and 3 carbon atom. So, the net result will be this 6 carbon atom, this 6 carbon atom and this 3 carbon atom. So, this becomes non-oxidative phase. Very important. Okay. So, if you simply see this diagram, because in this diagram it is little bit complex, so I just want to simplify this. What you have to remember is, जैसे In oxidative phase, we know ribose 5-phosphate is produced. Okay. So this ribose 5-phosphate will be converted into xylulose because they are linked as isomers. So isomerase will be the name of the enzyme. But the main thing which you have to remember here is that this 5-carbon atom combines with a 5-carbon atom and it splits up into 3-carbon atom and 7-carbon atom. Okay. This 3-carbon atom is obviously glyceraldehyde and 7-carbon atom is pseudoheptulose 7-phosphate. Then this 7-carbon atom again combines with 3 carbon atoms, again 10 carbon atoms, and it splits up to form 6-carbon atom and 4-carbon atom. Why I have not circled 4-carbon atom? Because 4-carbon atom will combine with 5-carbon atom to form a 6-carbon species and 3-carbon species. So I can say 6 is fructose 6-phosphate, fructose 6-phosphate, and this is 3-phosphoglyceraldehyde, right? And we all know that this entire thing will merge into glycolysis, right? This is what you have to remember. And that is what is given in this picture. Okay, you can see that xylulose 5-phosphate, ribose 5-phosphate, 5 plus 5, 10. It is forming pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate. Again 7 plus 3 is 10. Again you can say 6 and 4. Okay. Then this 4-carbon atom is again combining with a 5-carbon atom species to form 6 and again 3. So the net resultant will be this, will be this, will be this. That is what is explained in the previous picture. So it's up to you. If you can understand through this, it's well and good. If you can understand by that one, you can go through that. This is nothing but this is what is your, this is oxidative pathway, which I have explained separately also, and this is your non-oxidative pathway or phase. Okay. Now the question is, you have to remember the enzymes, because again I am telling you, without enzymes it is very difficult to understand the things. Now the enzymes here are, the enzyme is transketolase. This is a very, very important enzyme. Transketolase. Always remember, this enzyme requires TPP as a coenzyme. TPP and magnesium ions are required. Okay. Here the enzyme is trans-aldolase, and here the enzyme is again transketolase. Again TPP. So I can say that in non-oxidative phase there are 2 transketolase reactions and 1 trans-aldolase reaction. Right? So in non-oxidative phase 2 transketolase reactions, and in this 1 trans-aldolase reaction takes place. As you can see here also, TPP, transketolase, trans-aldolase, and again transketolase. So this is HMP shunt, right? This is very, very important. Now after this HMP shunt, then obviously we need to know what is the significance of HMP shunt. What is the use of this? So if we talk about the significance, as you have seen its significance, that it is producing NADPH. So the first important feature of this is it produces NADPH, and you have to remember it as NADPH, not NADH, because if you write NADH, your answer will be wrong, because NADPH is having high energy phosphate, and you will see the use of NADPH in lipid metabolism, in fact protein and amino acid metabolism. So the first feature is it produces NADPH. Now the question which might strike your mind is, what is the use of NADPH, isn't it? So if we talk about in general, what is the use of this NADPH? In steroid synthesis. When we have to make 6 hormones in the body, NADPH is required. In lipogenesis or synthesis of fats. In the synthesis of cholesterol. So many uses are there, and in amino acid metabolism also. Right? So NADPH, that is why I told you that it plays a very important role in adipose tissue. So again, this demarcates the use. And along with this, the most important role played by this NADPH is it maintains the integrity of cell membrane of RBCs. Very important, which we will be discussing again in detail after this. Membrane of RBCs, right? And second important feature is that it produces ribose sugar also. Produces ribose sugar. Ribose, which is a pentose sugar. And what is the significance of this ribose sugar? That it is required for the synthesis of the bases of DNA. So I can say that produces DNA bases, or whatever you can say, purines or pyrimidines. To be more specific, purines and pyrimidines, which are the bases of DNA and RNA. Okay, and just imagine, since this is the most important feature of HMP shunt, and that is why it is operative in the body, because this is the only pathway which can produce NADPH and which can generate ribose sugar. Right? So this is the significance. Then coming over to this, the second point about this HMP shunt is that what role this HMP shunt plays in maintaining the integrity of RBCs. So next topic is, you can say, role in RBCs, which is actually very important. Okay. Now, if we talk about its role, just now I told you, क्या करता है? It maintains the integrity of that cell membrane. So it maintains the integrity of the cell membrane. Now the question is how it maintains the integrity. Okay, so what happens, because it is producing NADPH? So what is happening is, in our body, these free radicals, these antioxidants play a very important role. In general, if I have to, just in a layman's language, what is a free radical? Free radical माने जो free है. Free का मतलब क्या है? Free है, या you can say kind of he or she is ready to mingle with any electron. So the moment it mingles with any electron, it is going to make a friend. It is going to make a bond between, friendship kind of bond. So what will happen? It tries to attack the cell membrane, or it tries to attack the electrons present in the cell membrane. So in that bargain, these free radicals start eating that cell membrane. Okay. Up in our body, there is a special kind of defensive mechanism which will not let this bond to form. So it will prevent that. So usko hum kya bolte hain? We call them as antioxidants. So in general, I can say antioxidant is the one which will prevent the oxidation. Oxidation means which will prevent the formation of that particular bond. Right? So there are so many important mediators, messengers, or you can say the substrates present in our body which are functioning as antioxidants. Okay, so glutathione is one of the most important antioxidant present in the body, which actually plays a role in RBCs also. Now the question is, what is this glutathione? So this glutathione is actually a tripeptide, which we will discuss again, which are going to discuss in the metabolism of the amino acids. But here, for your knowledge, I'll just write one thing, that glutathione is an antioxidant. This much you have to remember for this HMP shunt. Antioxidant formed of 3 amino acids. So it has glycine, it has cysteine, and it has glutamate. Or commonly we call it as GCG. Okay. So obviously it is an antioxidant. It is going to occur in 2 forms. And what are those 2 forms? One is, this is oxidized form, right, and this is a reduced form. And this reduced form, SH, is coming from sulfhydryl group of cysteine. So it is like, which is giving it a reducing nature. Sulfhydryl group. Right. Now see what is happening. It is occurring in 2 forms. To convert this oxidized to reduce, you can see hydrogen is coming. This hydrogen is actually provided from NADPH. So there is a complete cycle which is going on. So this is the oxidized form, GSSG. This is the reduced form, GSH. So this is being converted into this. Simple reaction is there, reduction. अब reduction है, so the enzyme has to be reductase, and here NADPH will be there, and here simple you can say NADP positive will come. Right, now, and this will be converted back to this. So because there is a kind of cycle going on. अब since reduction is occurring, so the enzyme here is glutathione. Because we are reducing glutathione, so the enzyme has to be glutathione reductase. Okay, and one thing, glutathione reductase requires B2 as a coenzyme. As we have already discussed the role of coenzymes in the video on enzymes. So you know coenzyme, means the substrate which is working in cooperation with the enzyme. So NADPH will be converted into this and glutathione will be reduced. Now when glutathione is in its reduced form, it has to go back, because whatever is there, if oxidation is there, reduction has to be there, and that is what you have studied in chemistry also. Our body consists of typical redox reactions. If something is gaining electrons, the other has to lose, or if something is losing electrons, the other person should be there to receive those electrons. This is nothing but redox reactions. Okay. Now, in this Pargen, what is happening is, when glutathione is converted back to its oxidized form, H2O2, which is a very, very important, I would say, you know, the role of hydrogen peroxide. You know, hydrogen peroxide, वैसे भी, even if you go for manicure, pedicures, even in so many important things, hydrogen peroxide is used. Why? Because this is functioning as very, very important generator of free radicals also. Okay. So this H2O2 will be converted into water and oxygen. And again it will be converted back, because peroxidases, catalases, again they are the reactions. So you know, everything is a typical redox reaction. And if we talk about this reaction, the enzyme here is glutathione peroxidase. And glutathione peroxidase requires selenium as a cofactor. You know what is the difference between a cofactor and a coenzyme? Cofactor is the one which is a metal ion or a mineral attached to an enzyme, and coenzyme is the organic molecule which is attached to an enzyme. Okay, so this is the complete cycle which is going on the cell membrane of RBCs. And what is the source of NADPH? Just now we studied HMP shunt. And what is the name of the enzyme? By far you people should be familiar. I am writing it G6PDH. I am not writing the full form. We know glucose 6-phosphate dehydrogenase. Now see, HMP shunt is going on. G6PDH is there, which is producing NADPH. NADPH is converting oxidized glutathione to a reduced form. and in the presence of glutathione reductase, and this reduced form is leading to the breakdown of peroxide radicals. This is the process which is taking place in the cell membrane of RBCs. Now the question is, what is the clinical correlation of this? What is basically happening now? Now suppose if I say that this G6-PDH is not working. It's an enzyme. Okay. So suppose if I say G6-PDH is not working. What will happen? This reaction will be blocked. This reaction will be blocked. ठीक है ना? Because the one which is generating NADPH is not being produced. If NADPH is not being produced, glutathione will not be converted into its reduced form. If glutathione is not converted into its reduced form, there will be increased concentration of this hydrogen peroxide. That is what exactly happens. What happens is that in G6-PDH deficiency, this is very, very important. This is actually X-linked, and I will tell you how it leads to hemolysis. What will happen is, if G6-PDH is deficient, there will be increased concentration of H2O2. Now this H2O2, poor chap, it cannot be converted into H2O and oxygen, so it will be converted into free radicals. That is what I told you, free radicals, because you have studied again in chemistry, chain initiation is there, chain propagation is there, chain termination is there. Whenever a chain is started, it's like a trigger. When you start a chain, it will start multiplying at a faster rate. And the antioxidant which is there to terminate that, it is also absent because it is not working. Why it is not working? Because NADPH is absent. Okay. So H2O2, what will happen is, it will increase the production of peroxide free radicals. So there will be a fight now, and these free radicals will start damaging the cell membrane. Ab RBCs ki membrane, if it is damaged, it leads to hemolysis, splitting. So it will lead to hemolysis, and whenever hemolysis occurs, automatically it will lead to reduction in the count, and it will lead to hemolytic anemia. So you can see that a single mistake in the body, or a single absence of a single enzyme in the body, can lead to so many things. And you will be surprised to know that there are certain diet is there which also promotes its deficiency, that which we are going to discuss now. So it will lead to hemolytic anemia. Okay, so this is just a chain, that deficiency of G6-PDH will lead to hemolytic anemia. Very, very important. Now the question is, what is this? That means everything revolves around this G6-PDH. So always remember one thing, and that is why we say that the rate limiting enzyme, the rate limiting enzyme of HMP shunt is G6-PDH. It becomes the rate limiting enzyme also. Okay. And deficiency leads to, just now I told you, deficiency of G6-PDH will lead to hemolytic anemia. Right? And third important point is that the most important one, in fact, there is formation, you can say there is presence of Heinz bodies also in the blood. Okay. So the main feature of hemolytic anemia, the main is Heinz bodies present. Very, very important. And this is mainly due to the oxidative stress. Okay. And it is aggravated. Aggravated means it is increased. Right. When we have fava beans. Fava beans, this is a kind of, dish. You can say it's a kind of vegetable which aggravates this process. So aggravated by ingestion of fava beans. okay, favism, and then it is also aggravated by the oxidative drugs like जैसे primaquine and all. Okay, and it is aggravated where जैसे Mediterranean areas where there is Plasmodium falciparum. So Mediterranean areas also. Of Plasmodium falciparum, the one which is responsible for malaria. Okay, so it is aggravated by these three things, and this is strictly X-linked, means males are more prone to this G6-PDH deficiency. This is again a very important feature. So deficiency, it is X-linked. So only in males. Okay, so this is a very important alternative pathway of oxidation of glucose which is aggravated by these factors. And hemolytic anemia के लिए there are three other reasons also which leads to hemolytic anemia. जैसे we have discussed again in our previous videos, जैसे I told you that pyruvate kinase. You know now what is pyruvate kinase? It is a very important, I would say, a step of substrate level phosphorylation in glycolysis. Okay. So pyruvate kinase deficiency also leads to hemolytic anemia because glycolysis will be affected. PFK also leads to hemolytic anemia. Again, glycolysis will be affected. But the most important difference between hemolytic anemia caused due to G6-PDH deficiency and pyruvate kinase and PFK is no Heinz bodies will be present in PK and PFK deficiency. But here, if you take the blood smear, there will be presence of Heinz bodies 120% sure. Okay, because here it is affecting the entire sequence of the reactions. Okay, so this is the most important point which I will highlight also, that deficiency of G6-PDH leading to hemolytic anemia, X-linked, Heinz bodies present mainly due to oxidative stress, and these are the factors which are increasing that. Okay, and another important feature related to this is, ये तो हो गया, this is we have discussed about the entire cycle या pathway you can say which is occurring on the RBCs. Now I told you about transketolase. Okay, so what is the importance of transketolase, or you can say correlation. This transketolase enzyme is having TPP as a coenzyme. Okay, just now I told you as TPP as a coenzyme. So this is a measure to detect the deficiency of B1. So it's like a measure to detect B1 deficiency in RBCs. Very important. Okay, वैसे तो we can we can check the thymine excretion in urine also, but this is a very important step. What happens is, because transketolase is requiring TPP, so definitely if B1 is deficient, there will be a decrease in erythrocyte transketolase activity. We call it as erythrocyte transketolase activity. Very important. Okay. Second important feature here is, one is this transketolase. Second one is, you can say, glutathione reductase. Glutathione reductase, which requires B2 or riboflavin. Okay. So I can simply say, what is the significance? That erythrocyte glutathione reductase activity will be a measure for activity will be a measure for B2 deficiency. Measure for B2 deficiency. These two are very important ones. And the third is, it is like important only, glutathione peroxidase requires selenium as a cofactor. So, what is selenium? Selenium is acting as an antioxidant. We know that selenium is a trace mineral, functions as an antioxidant. So, these are the important points about this HMP shunt, which is generating NADPH and ribose sugar. NADPH is required for, I told you that for variety of reactions, it has a very important role to play in the integrity of RBCs, and second is ribose sugar. Ribose sugar is required for the synthesis of purines, which again we are going to discuss, which will be discussed in the mol bio videos. Okay. So whether it is adenine, whether it is guanine, the precursor of purines and pyrimidines in the body is none other than ribose 5-phosphate, and this ribose 5-phosphate is coming from HMP shunt, and that is why this HMP shunt is important for the body, which is an alternative pathway of oxidation of glucose. So this is all about the HMP shunt. Then after HMP shunt, then we come over to the second alternative pathway of oxidation of glucose, which is uronic acid pathway. Now see what happens in uronic acid pathway. It is again an alternative method, right? Uronic acid pathway. Now, एक तो it is not a shunt, ठीक है, it's a pathway. So I can say that site, the most important site of uronic acid pathway is the liver. Main is the liver, right? Now the question is that, chalo, the site is liver. What is the significance of this? The most important significant feature of uronic acid pathway is that this pathway produces glucouronic acid. Produces glucouronic acid. Not gluconic acid, it is glucouronic acid. Now, what is the significance of glucouronic acid? Okay. So, the most important role of this glucouronic acid is that it plays a very, very important role in the degradation of heme. Very important. So, the significance is it produces glucouronic acid. Now, what is the significance of glucouronic acid? So, it produces glucouronic acid which helps in the degradation of heme. So, heme degradation. And what reaction? It plays a very, very important role in conjugation. So this is one important feature of glucouronic acid, and second important feature is in the synthesis of mucopolysaccharides. Right? So these are the two features, very important features of glucouronic acid, and you will be surprised to know that वैसे भी you are going to study in medicine that absence of this glucouronic acid is responsible for different type of jaundice. It is responsible for causing so many different syndromes in the body. Okay, so you can just imagine the importance of glucouronic acid, and the only pathway which can produce glucouronic acid in the body is uronic acid pathway. So you can just imagine the significance of this, right? Now the question is, ये तो हो गया in human body, how glucouronic acid is produced by uronic acid pathway. Then in non-primates, okay, non-primates में uronic acid pathway leads to the production of vitamin C also. in non-primates. This is also an important topic which is generally asked in the exam, that uronic acid pathway produces vitamin C, or you can say produces ascorbic acid, because we won't say it as a vitamin because it is not provided as a supplement in non-primates because it is already synthesized. But yes, in human beings ascorbic acid cannot be produced because uronic acid pathway is operative till the formation of glucouronic acid. I'll show you the pathway also after this. So I can say that we human beings cannot produce ascorbic acid. So humans cannot produce ascorbic acid. Ascorbic acid, why? Or I would say that is why it is also known as vitamin C, because vitamin C is a very important vitamin as an antioxidant, and so many other uses are there. So humans cannot produce ascorbic acid as we lack this enzyme. The name of the enzyme is L-gulonolactone oxidase. Since we lack L-gulonolactone oxidase, we cannot produce ascorbic acid, and this uronic acid pathway is till the formation of glucouronic acid. And I'll just show you the pathway also. What happens here is, this is a very interesting pathway, you can see that. Okay, simply we start with glucose, and here you can see that glucouronic acid is produced, right? And here if we talk about, you can see that once glucouronic acid is produced, it is required by again heme degradation, conjugation reactions, and it is used further. And you can see that glucouronic acid is then converted into xylulose 5-phosphate, and this xylulose 5-phosphate can enter HMP shunt. Again same thing, it is an alternative method, because starting say I am telling you that alternative pathways are actually restricted to particular organs. Why? Because they are giving rise to certain useful compounds, and once they form those useful compounds, they merges themselves into the major pathway. Ab this xylulose here, so you can see that chalo, it is merging into HMP shunt also. This xylulose 5-phosphate can be converted into ribulose or ribose 5-phosphate. Again it can be used. Okay, right. So this is what is uronic acid pathway. Now another important feature in this pathway is that obviously if something is produced, it is merged also. But what is the clinical correlation? The clinical correlation is that if there is absence of xylulose reductase, as you can see that this enzyme is degrading xylitol. So if this enzyme is absent, what will happen? There will be increased concentration of this xylulose, which is a pentose sugar, in urine, and it will lead to essential pentosuria. But yes, it is a harmless condition. Okay, so I can just simply write it like this, that what is the clinical correlation? We can say that correlation is that excretion of xylulose 5-phosphate in urine. Why? Due to the absence of enzyme xylitol, or you can say xylulose reductase, right? And this is also known as essential pentosuria. So these are the two alternative pathways of oxidation of glucose. Now let's quickly summarize that what are the major pathways and what are the alternative pathways. So if we talk about the major pathways, or if we talk about the alternative pathways, right? Under major pathways, you start from the first one, glycolysis, right? Gluconeogenesis, glycogenesis, glycogenolysis, right? Then you can say TCA. TCA is major, or you can say it is amphibolic also. Okay, these are the major pathways. And if we talk about the alternative ones, HMP shunt, we will always write it as shunt only, uronic acid pathway. Right. Up now the question is that major pathways are also there, minor pathways are also there. We are having, along with glucose, we are having galactose, we are having fructose also in our diet. So galactose and fructose, when we are having them in our diet, how they are oxidized? So they will be alternative, or I would say they are the minor pathways, because along with that we are eating other things. So what are the minor pathways? Under minor pathways, we are going to study the metabolism of galactose. So we can say galactose metabolism will come, and fructose metabolism will come. Right. So in our next video, we are going to study these minor pathways. I hope these alternative pathways of oxidation of glucose is clear to all of you. Thank you so much.